Okay, hi everyone. This is Lecture 17, Blood Vessels and Blood Pressure. So today we're going to continue the cardiovascular system. We're going to review the structure and the basics of the individual blood vessels, and then we're really going to do a lot of equations today. So make sure you have some extra paper, make sure you've got some coffee, and put on your game face and try not to be intimidated by all the equations we're going to throw at you today. We're going to go through them slowly. We're going to talk about why they're important, and we're going to look at various measures in the heart for how well the heart is functioning as it tries to get blood out to the tissues. So we're going to talk about blood flow and blood pressure through the different blood vessels: arteries, arterioles, capillaries, and veins. And then at the very end, we'll bring it all together and I'll give you a summary of regulation of the heart and the blood vessels. So just to remind you, the previous lecture focused on the heart. So we talked about the heart as a double-sided pump. And as the heart is pumping, it's establishing blood pressure that is needed to get the blood flow out to the tissues. The blood vessels then are passageways for the blood to be distributed throughout the body so that the blood can exchange materials to and from the tissues. The blood itself then will be the subject of our next lecture, which is a liquid connective tissue that actually transports the dissolved and the suspended materials that are going to be exchanged to and from the tissues. So the focus of today is really about circulation. So we learned in the previous lecture how the heart pumps blood through the contraction of the ventricles. It's going to pump blood either out the pulmonary trunk to the lungs or out the aorta to the body. So as the heart is pumping, it's establishing and increasing blood pressure to get that flow through the blood vessels. And as the heart is relaxing, it's receiving blood to refill to reset the circuit. So the heart and the blood vessels form a closed circuit in which the blood pressure builds up based on the pumping of the heart. And that allows blood to flow throughout the system. Just like any other closed circuit that involves fluid dynamics, any break in the circuit or leakage of fluid can seriously damage the ability for the blood to get out to the tissues. In addition, we need to have a functioning pump. The heart has to be working appropriately in order to get enough blood pressure established such that the blood actually reaches the tissues, and enough blood pressure such that it is continued through the circuit so it gets back to the heart and recycled. So we really want to think about, as we go through all the different measurements today, we want to think about, is the body getting sufficient circulation, and how would we measure that? So the heart needs to pump adequately to get enough blood to the blood vessels, and we're going to measure that in cardiac output. So cardiac output is how much blood the heart is pumping out. The blood vessels then will receive and respond to changes in pressure to adequately get blood to and from the tissues. So we have some blood vessels that are more elastic that can help to recoil and push the blood through. We will have some blood vessels that are less elastic and more open that are going to be pressure reservoirs, decreasing blood pressure. And then we will have some vessels that can actually be changed by the system. So how dilated or how constricted they are, how open or how closed they are, can then determine which tissues will get fluid, and also can determine overall in the whole circuit what the pressure is in the whole circuit. So we will talk about blood flow to individual local tissues, and then we will talk about blood pressure in each of the different blood vessels locally, and then the entire systemic blood pressure, which is called mean arterial pressure. So blood vessels are responsible for transportation of blood from the heart to the tissues, and from the tissues back to the heart. And in general, it's like this. Arteries move away from the heart and towards the tissues. Then arteries branch into smaller arterioles, and arterioles branch into even smaller microscopic blood vessels, which are capillaries. Those microscopic blood vessels then exchange materials with the tissues, and we will talk about that exchange in capillary bulk flow. And then from there, they will pick up materials, pick up wastes from the tissues that needs to go back to the heart to be removed by the lungs. And that occurs through the smaller blood vessels, the venules, and then to the larger returning blood vessels, the veins. So venules and veins move towards the heart, returning blood from the tissues. So let's draw this out quickly so you guys can think about this flow. Okay, so we have the heart, which I'm going to draw just as a very simple heart. The left side of the heart is going to pump blood away towards the tissues. The right side of the right side of the heart is also going to pump blood away towards the lungs. So these are our pulmonary circuit, where blood gets pumped away from the heart to the lungs, and then our systemic circuit, where blood gets pumped to the body tissues. So the largest blood vessels that move away from the heart are the arteries. And the arteries are going to branch into smaller arterioles. And then from there, once we reach a specific local tissue, we're going to get microscopic capillaries. The capillaries then is where the exchange is going to occur with the tissues. And then the blood returning to the heart will go to the smaller blood vessels towards the heart, the venules. And then from the venules to the veins, the larger blood vessels, the last of which will then enter the heart and return blood to the heart. We could do the same for the lungs, where we have arteries, arterioles, capillaries, venules, and then veins. And those will then return to the left side of the heart. So we will talk more when we get to the respiratory system about the actual exchange of oxygen and carbon dioxide and waste that occurs. But for now, I want you guys to just think about the blood flow and the actual components of the blood vessels. So we're going to go one by one through these. We're going to start with arteries. So arteries move blood away from the heart towards the tissues. If you remember back to your anatomy, arteries have thick elastic walls and a very large radius, or they're very large vessels. They are very high pressure vessels because they are closest to the heart as the heart is pumping blood out. The large elastic arteries will expand like balloons to absorb and store pressure of the pumped blood leaving the heart, and then they will recoil, pushing the blood forward to the next vessels in the line. So for that, you will see within the artery walls, you will see a lot of tensile strength. So you're going to see collagen on the outer connective tissue for the strength of these large vessel walls. You will also see elastic tissue. And these elastin fibers allow that stretch and recoil so that the pressure in these large arteries will fluctuate with the diastolic and systolic events in the heart. So as the heart is pumping, the arteries will expand to receive that blood, and as the heart is relaxing, the arteries will recoil to push the blood forward. From the arteries then we go into the smaller vessels away from the heart, and these are arterioles. Arterioles are highly muscular. They have very dense sympathetic nervous system innervation. So shown here, you can see that there is a lot of smooth muscle in the arterioles. They are very nicely rounded vessels. with a smaller radius than the larger arteries. They do have a connective tissue layering, but they do not have any elastic fibers like you see in the large arteries. So they are primarily smooth muscle. So the arterial smooth muscle can contract to reduce or constrict the arteries. And that is going to be used to regulate blood flow. And we'll talk about what happens when you constrict arterioles either locally or systemically. So this is a thick, circular, smooth muscle layer within the arterioles. So when the sympathetic nervous system excites the arterioles, you will get vasoconstriction. So you're going to have sympathetic nervous system input, which is going to release what neurotransmitter, guys? Epinephrine. It's going to release epinephrine and norepinephrine to the arterioles, and what that does is it constricts or tightens the arterioles. That will decrease the diameter of these vessels. When the sympathetic nervous system is lower or less, when that epinephrine is at a lower level or not being released, then the arterioles will dilate and they will be more relaxed with a larger diameter. So this is called the vasomotor response. It can be triggered in any vessel that has smooth muscle, but it's primarily triggered in the arterioles. And it has the greatest effect in the arterioles because they are small diameter. So changing something that is small diameter to a smaller diameter has a huge effect on the pressure within that arteriole. From there, we get to the microscopic blood vessels that actually are the sites of exchange with the tissues. These are very thin-walled, really just a single layer of endothelium, and they're very low pressure to slow down the blood flow to allow the exchange to happen with the tissues. Their permeability can vary depending on the location in the body and how tight the endothelial cells are joined together. So here are the endothelial cells forming a capillary. And based on this, we have different types of capillaries. Capillaries that are very open, that contain almost pore-like windows, are fenestrated capillaries. Fenestrated means windows. So they have large pores. Continuous capillaries are then sort of the general capillaries that we have in the body that are not very permeable, but they do allow for exchange because of their very thin walls. And then the tightest junction capillaries, which are surrounded by neuroglial cells in the brain, form the blood-brain barrier. So these are just endothelium, no elastin fibers, no smooth muscle, and no collagen fibers. From there, then, we're going to do the exchange in the capillaries and then return blood to the heart. So the large vessels returning blood towards the heart are the veins. They have thin walls compared to the arteries, but they are still large radius. So large venules, and they actually do have some smooth muscle, sympathetic nervous system innervation. Not as much, but they can respond to sympathetic nervous system, and they can constrict and tighten when the sympathetic nervous system is active. They also have valves, and I'll show you those in a bit, but they ensure that even when the veins are low pressure, that the blood is slowly moving towards the heart and not backfilling. They are easily stretched to retain the blood, but they are not elastic, so they do not recoil. Very little elastic tissue. They are slow flow and low pressure vessels. So because they are slow flow, because they are large, the veins at any given time in the cardiac cycle, at any given time in the body, contain about 60% of the total blood volume. That's because they are the blood is kind of pooling in the veins. The venules then are just the tiny veins that are just leaving the capillaries. They are very thin walls, very small radius. So here is a picture that shows how much blood is in the various portions of the cardiovascular system, and you can see that about 64% of blood hangs out or slows and pools in the systemic veins. The rest of it is moving through, pumped from the heart through to the rest of the arteries and arterioles and capillaries. So this is a summary diagram for your reference to compare large arteries with their elastic tissue, arterioles with primarily smooth muscle tissue, capillaries with primarily endothelial tissue, and then the large veins, which have smooth muscle but very little elastic tissue. So again, let's get back to this idea of circulation. So the heart is pumping blood out the large arteries, and then it's going to travel through the arterioles and then to the capillaries. At the capillary level is where the exchange is going to occur with the tissues, and then the tiny venules will bring the blood back towards the heart and merge into large veins before it completely returns back to the heart. So we're going to then measure several things in the blood vessels to see how effective the delivery of blood to the tissues is and how we can change that based on conditions. So you can imagine that sitting quietly, you need a certain amount of blood pressure and blood flow compared to, say, exercising, where you need to increase your blood flow to the tissues to get more oxygen and remove more carbon dioxide. So the flow rate is the volume of blood passing through a vessel in 1 minute. So how much blood is getting through the vessels in a single minute? That's the flow. The resistance is opposition to that flow. So there is friction as the blood moves through a vessel, and the resistance is the term that accounts for that resistance. So we have the blood moving through, and then we have the resistance, which is stopping the blood from moving through. So they're opposing forces. We can then measure the speed of the blood flow, and we will also measure the force of the blood, how much pressure is pushing against the vessel walls as the blood moves. So arteries first are pressure reservoirs. So what does that mean? That means that due to these elastic properties of the large arteries, so they are stretching to receive blood as the ventricles pump, and then they recoil to push the blood to circulation. What that means then is that the ventricle pumps, right? It pushes blood into the arteries and the arteries stretch to receive that blood. Then the ventricle is going to relax, and the ventricle pressure is going to get very, very low. But at that same time, the arteries are going to be recoiling to push the blood out. So this ensures that at no point in time in the body does the total blood pressure go down to zero. Even as the heart is relaxing and becoming very low pressure or close to zero to fill, the arteries recoil to continue pushing the blood forward. So these pressure changes reflect then the cardiac changes. And we can measure pressure in vessels by looking at systolic and diastolic pressures. So the maximum pressure occurs when the ventricles pump the blood out to the arteries. The minimum pressure occurs when the ventricles relax. The maximum pressure, because it's during the contraction phase, is the systolic pressure. The minimum pressure, because it occurs during the relaxation phase, is the diastolic pressure. So because the arteries are large, they are also very low resistance, meaning that there's not a lot of opposition to the flow. So the arteries are going to be able to faithfully receive and push the blood forward without any opposition to that. So here's just a view of those elastic fibers, and this is the aorta, which is the largest of the arteries. So these are the elastic fibers that are allowing this stretching and recoiling of the aorta. And here's a picture from your book, which is maybe better than my hands, right? So here is blood returning to the heart, filling the heart, and now the ventricle's contracting. When the ventricle contracts and pushes blood out, this elastic large artery like the aorta is going to receive that blood. And then when the ventricle relaxes, goes down to low pressure, the whole system could go down to very low pressure, but because the artery then recoils, it pushes blood as the ventricle is relaxing, and that continues to maintain pressure through the system. So if you don't have this recoiling of the arteries, you can have dangerously low blood pressure during the relaxation or diastolic phase. So now we're going to do some measurements and talk about some of the major components and regulation of blood pressure. So systemic blood pressure is measured at the arteries because of their elastic properties. So the question here is, how strong is the movement of blood through the circulatory system? And why do we care about this? We care about this because ultimately what the systemic blood pressure tells us is the heart effectively pumping enough to get blood through the system? So a couple of definitions here, and we're going to measure blood pressure in lab as well. So blood pressure is the force exerted by the blood against a vessel wall. So that is how much the blood is pushing against the vessel wall. This depends on how much blood is present and how much the vessel walls can stretch and recoil the compliance and distensibility of those vessels. We can measure blood pressure locally in a single artery, or we can measure blood pressure systemically in the entire system. And if we do that, we call that mean arterial pressure. And that is the pressure in the arteries that determines the total pressure in the entire circulatory system. So you can imagine there's different times when we would want to do this, right? Sometimes we want to know locally how much blood is getting to a certain area. Let's say you're doing a lot of squats and you're working your legs a lot. You're going to get some excess blood going to those tissues that really need it. But sometimes we also want to think about the total system. And in that case, we're looking at mean arterial pressure, and this will be a calculation based on the difference between systolic and diastolic events in the heart. What we use to measure blood pressure is a blood pressure cuff, and the fancy term for that, see if you can say this 10 times, sphygmomanometer. So the sphygmomanometer is that thing that they put around your brachial region or your upper arm, and it's a blood pressure cuff used to measure changes in arterial pressure. So if we look at pressure through all of the arteries, okay, and I know this is a bit of a confusing graph, but take a second to look at this. So here we have pressure, which is measured in millimeters of mercury, from 0 to 120. The left ventricle, look at how much the left ventricle varies. So when the left ventricle is relaxed, it goes all the way down or very, very close to 0. When the left ventricle pumps, look how strong, all the way up to 120 millimeters of mercury. So this is the pumping, filling that is happening in the left ventricle. Now we don't want this big of a drop to be throughout the system. That's not good for the tissues to have these huge deviations in pressure. It's also damaging to have such big spikes in pressure. It's okay for the heart is built for that, but once we get to the vessels, we need to then smooth that out. So the large arteries are going to receive that high pressure, that 120, and then they're going to recoil to make sure it doesn't go all the way back down to 0. So the large arteries are going to be usually on average between 120 max during contraction or during the systolic events, and 80 minimum during the diastolic events. So this is where your blood pressure measurements in the arteries, for example, the brachial artery measurement that's done with the sphygmomanometer is going to be 120 over 80. not 120 over 0, okay? Because that's not healthy for the rest of the system. So 120 over 80 is a typical systolic diastolic pressure. And then you can see as we get down to the other vessels, the arterioles, the capillaries, the venules, and the veins, the pressure goes down. So in the arterioles, we have more fluctuation, but as we get further and further away from the heart, the pressure goes down and the fluctuations disappear. We can measure the mean arterial pressure rather than the systolic and diastolic fluctuations by calculating how much blood pressure is through the system as a combination of systolic and diastolic, and that's our mean arterial pressure, which is this smooth line going through the system. Okay. So I want you guys just to be kind of familiar with these values. So systolic pressure, 120 over 80. Mean arterial pressure, usually somewhere around 90 to 95. Pressure in the arterioles is going to go from around 90 to the very smallest of arterioles to about 40. By the time you get down to the capillaries, we're in the 30 to 10 millimeters of mercury range, and then in the venules and veins, nearly dropping down from 10 to 0. Not totally 0 because we need enough pressure to get back to the right atrium. Okay, so measuring blood pressure. The highest pressure during ventricular contraction is the systolic pressure. The lowest pressure during ventricular relaxation is the diastolic pressure. So remember, this is about 120 for systolic and about 80 for diastolic. The mean arterial pressure then is the average pressure in the system, reflecting how much blood is actually getting delivered to the tissues. So as the values are fluctuating, what ultimately actually gets through is the mean arterial pressure. And we can calculate that partly by looking at the pulse pressure, which is the difference between the systolic and diastolic pressure. So here, if we just look at the fluctuation in the arteries now, we're just zooming in on this portion of the graph, so just the arteries, and this is what it looks like. We're going to have 120 over 80, so 120 for systolic, 80 for diastolic. The difference between systolic and diastolic will be your pulse pressure. Then you will have the mean pressure, which is a calculation I'll show you in a minute, which calculates how much is actually getting to the tissues. You'll have a little notch here, interesting enough, that is caused by the closure of the aortic valve as we prevent backflow from the aorta back to the ventricle. So this is what a sphygmomanometer looks like. It's this pressure cuff around the arm. And when we measure blood pressure, this is what the system will look like, okay? So I'm going to leave this for your reference after we go through the blood pressure lab and describe all of this, and you guys have some experience listening to and thinking about the blood flow through the brachial artery during this blood pressure measurement. Come back to this slide, okay, after we do the lab. Okay, now let's get to the equations. So, I know, are you excited? So get some paper out, guys. There's going to be several equations, and I want you to keep track of them as we go through. Okay. Before we get to this mean arterial pressure one, we had a couple of equations from last time. Do you remember them? Nope? Yes? We're going to go through them really quick, okay? Because I want you guys to remember them so we can keep track of where we are. Okay, so last time we talked about the cardiac cycle. So the cardiac cycle was dictated by the electrical and mechanical events in the heart. So in the cardiac cycle, we basically had filling events and pumping events, right? So the filling events are the diastolic events, and remember we're referring to the diastolic events of the ventricles. And the pumping events are the systolic events. Remember, that's systolic of the ventricles. So what we had last time, just to review the cardiac cycle, we initially started out as blood, oops, I should draw that in blue, sorry guys. Blood is filling the heart, and deoxygenated blood is filling the right side, Deoxygenated blood is filling the right side, oxygenated blood is filling the left side. And we're going to hit a point where both sides of the heart, both ventricles, are maximally filled. That max filling of the ventricles is going to be your end diastolic volume, or your EDV. This is the max fill. Does anybody remember the final event that causes the max filling of the ventricles? That is when the SA node becomes active and actually contracts the atria. So the final push to get the last bit of blood into the ventricles and reach EDV is when the atria contract, and that's caused by the SA node. Okay, so our EDV is our max fill. And then from there, the electrical activity is going to spread down the ventricles and cause the ventricles to contract. So when the ventricles contract, then we are in the pumping phase, or the systolic phase of the ventricles, so they're contracting. And when they're contracting, the blood is now leaving to the lungs, or leaving to go out to the body. At the very end of that contraction, you'll have a little bit of blood left in the ventricles, and that blood that is left is the end systolic volume, or the minimum fill of the ventricles. We can then calculate the stroke volume by subtracting the two. So how much blood got out? The stroke volume then is equal to the end diastolic volume minus the end systolic volume. So the stroke volume is the end diastolic volume minus the end systolic volume. And we can then use that stroke volume as one of our measures of how much blood is getting out from the heart to the tissues. So that measure, which uses stroke volume, is called cardiac output. So cardiac output is going to be how much blood is pumped out of the heart per minute. And we get the per minute by multiplying it by heart rate. So we're going to look at stroke volume and also heart rate control. So the total cardiac output then is telling us how much blood is getting out to the tissues. So now, in these measures for blood pressure, now you're going to see cardiac output come back. So here is the cardiac output, how much blood is getting out per minute, which is going to help determine the total pressure in the system. Then we have to not only look at how much blood is getting out, but we also need to look at the resistance to that blood movement, or the resistance to that cardiac output. So the blood is being pumped out, but now it's being received in vessels. And the blood that is being received in the vessels is actually going to be opposed based on how open those vessels are, based on various other measurements within those vessels. So this measurement here is called total peripheral resistance, or how much the blood vessels are stopping this flow. So we have the blood being pumped out from the heart, and the blood vessels, which are carrying the flow but can also get in the way of that flow. We can also calculate the mean arterial pressure, which we do clinically. So when we measure the diastolic and systolic blood pressure, this is a quick way to calculate out in the field what the mean arterial pressure is once you get those measurements. So this is the theory, cardiac output times the total peripheral resistance, and this is the clinical way to calculate it as well. So you can use either one of these calculations depending on what we're doing. So we're going to measure systolic and diastolic pressures and use this to calculate. We can then look at how the cardiac output and the total peripheral resistance, or the opposition to the flow, is affecting the arterial pressure. Again, this all goes back to how much blood is getting delivered to the tissues so that they can exchange materials to and from the tissues. So the first thing we need to talk about is blood flow. So the flow rate through a vessel is how much blood flows through a vessel at any given time. This is the volume of blood passing per minute, and it's going to depend on the pressure gradient in that particular vessel and the opposition to the flow, or the vessel resistance. So the blood's going to enter. There's going to be a pressure gradient that pushes it through the vessel. And then there's going to be some opposition based on how small the vessel is, other structural components of the vessel, which will get in the way of that flow. So we can measure blood flow by looking at pressure gradient and resistance. Blood will flow just like any other fluid from high to low pressure. This will be determined by the cardiac output and by the blood vessel stretch or compliance. The resistance is the opposition of the flow caused by the friction of blood moving inside a vessel. There's a couple of things that can affect resistance. We can affect the resistance by changing the thickness of the blood. So if blood is thicker, usually this occurs because you have more red blood cells. When the blood is thicker, it's more like molasses as opposed to water, then it's slower flow. We can also change resistance by increasing the length. So every time the blood bumps against the wall of the vessel, we get more and more friction and more resistance. So the longer you ask the blood to do that, the longer the blood vessel is in length, that can be more resistance. Also, a tighter space. So if you have a tighter space for the blood vessel, it's a smaller diameter and it's going to be more likely to bump against the wall. So if you have a large vessel, the blood bumping against the wall is not as much, but you have a small vessel, it's much tighter and much more resistance. So we can summarize this with our blood flow equations. So here we say that the flow rate is determined by the change in pressure, or delta P, and opposed by the resistance. And the mathematical way of saying that it's opposed by the resistance is to divide it. So the resistance reduces. Every time the resistance goes up, the flow rate goes down. So flow rate is increased by the change in pressure and decreased by resistance. The resistance then has another fancy equation. What affects the resistance? Well, viscosity, how many blood cells, or how much like molasses the blood is, the length of the vessel, how long is the blood vessel, and the radius. It turns out that the radius is the biggest determinant of the resistance. So rather than just being a straight radius, we actually have radius to the 4th power. So there is a 4-fold change in resistance every time the radius gets smaller. And the radius, or how small the vessel is, affects resistance in an opposing way. So the smaller the vessel, or the less the radius, the bigger the resistance. In other words, the bigger the radius, or the wider the vessel, the smaller the resistance. And that's why, again, we divide by the radius here. So let's look at how to actually apply this. Okay, so write down these equations. Flow rate is the change in pressure over the resistance. And let's look at these individual terms and how they change. So first, here's the change in pressure. So if we keep just the resistance totally the same across any given blood vessel, and all we change is the pressure in and the pressure out. Here we have flow rate is delta P over R, and we're going to keep R the same. And we're just going to look at the difference in pressure and how it affects the flow. So here's one blood vessel where the blood pressure at the first part of the blood vessel is 50, and it goes down to 10. So that delta P is 40 millimeters of mercury. In vessel number 2, we will then increase the pressure, but keep the end pressure the same. So this is a much bigger difference in blood pressure, where we start out with 90 and we go all the way down from 90 to 10. That's a delta P of 80. When we do that, it's a pretty simple calculation. We went from 40 to 80. So the change in pressure in vessel 1 is half the change in pressure of vessel 2, or the flow in vessel 2 will be twice that of vessel 1. Okay. Are you guys getting lost yet already? Okay, let's look at it again. You started out with a much bigger pressure in blood vessel 2, and that big change in pressure caused a big flow rate, right? You pushed harder on it, you ended up at the same pressure, big change in pressure, big increase in blood flow, okay? Now, let's make the delta P equivalent. So if we look at from 90 to 10, a delta P of 80, and 180 to 100, even though this is very high pressure, 180 to 100, the delta P is the same. The flow rate in these vessels will still be the same, okay? Because it's ultimately the pressure gradient, or the change in pressure, that matters to the total flow rate, okay? So the flow rate changes as delta P over R. So let's draw this, because I know this is getting confusing. Okay, so the flow is equal to the change in pressure over the resistance. Now, based on this, when you want to increase flow, you are going to decrease resistance. So if you want to increase flow locally, and this is going to change when we talk about the whole system, so make sure that you emphasize here locally. When you want to increase locally to a tissue, you want to decrease the resistance, or have a big, open, dilated vessel. So to increase the flow locally, you're going to dilate. And you will see when you dilate blood vessels that blood flow will move in, and those tissues will become reddened because of that increased blood flow. So if you exercise a lot, a particular muscle, like you do a bunch of squats, then your leg muscles are going to turn red because you've dilated, opened up those vessels, decreased the resistance, and allowed the blood to flow through. If you want to cut off flow to a tissue, maybe for example, to get more flow to another area, if you want to decrease the flow, then you will increase the resistance. So again, this is local to a tissue. You want to have a small or more closed, constricted vessel. So big flow, big vessel. That's going to get blood to a tissue. Small flow, small vessel. That's going to slow down or stop the flow to a particular tissue that is not working. So this is usually to active tissues, and this is usually to less active tissues. So the radius then effects on resistance are fourfold changes. So we have these big open vessels to tissues that we want to get a lot of blood flow to, and small tight vessels to tissues where we don't want to get a lot of flow to. And again, these are 16-fold changes. So every 1 millimeter change you have in a vessel, you will have 16 times the change in the blood flow. So it doesn't take a lot to get a big change in blood flow. And this is mostly done in the arterioles. Arterioles are very high resistance, and they are the major determinants of flow rate in the body. Because they don't expand and recoil like the arteries, their flow rate is determined mainly by resistance. They're small radius, and their radius is adjustable based on sympathetic nervous system input. So more sympathetic nervous system input, more constriction, tighter vessels. Less sympathetic nervous system input, large radius, dilate the vessels. But it's not always sympathetic nervous system input. Actually, we have a lot of local changes that can change the smooth muscle in the arterial walls. But let's just visualize this. So a very high resistance or a tight blood vessel is going to have little to no flow. A moderate resistance or medium-sized radius is going to have a moderate level of blood flow, and a low resistance or a nice open dilated vessel is going to have a big flow and get a lot of blood to those tissues that really need it. Again, there's pictures of the smooth muscle in the arterioles, and I already talked about sympathetic nervous system, but there are several local controls that tell us when a particular tissue needs more blood supply. So let's go through each one of these on the list. So first, there are metabolic changes. So you can have metabolic changes where a tissue is very active. So near this arteriole, if the tissue is extremely active, then you will have low oxygen, increased carbon dioxide, increased acid or lactic acid, increased potassium, increased osmolarity, and increased adenosine from the use of ATP. All of those metabolic changes that happen when a tissue is very active will cause opening or vasodilating locally to that specific tissue. That increases the flow, say, to your quadriceps when you do squats, right? To get more blood flow to the more active tissues. When you don't have those signals, or in the absence of those signals, you will have vasoconstriction, which will decrease blood flow to the less active tissues. This type of vasodilation, vasoconstriction, is most pronounced in the heart, skeletal muscles, and the brain. It can alter the distribution to various organs and tissues. So I want you guys to circle this. This is local. Changes at a particular organ or tissue level that will cause vasodilation to get more blood to that specific tissue. And various parts of the body can do this. So here's the changes before exercise and after exercise. So skeletal muscles are going to be very metabolically active during exercise. They're going to increase their flow rate by 1,000%. The heart, etc., will also, based on these metabolic changes, increase their flow. We can also have local paracrine release. So the most studied is nitric oxide. Nitric oxide is a chemical that is released in response to metabolic changes that will cause, again, vasodilation. There are some other less well-studied paracrines that can cause changes. Endothelin causes vasoconstriction. Vascular endothelial growth factor can cause new vessel growth. So these are local chemical changes that can affect the diameter of blood vessels, the radius of blood vessels. Histamines are also local chemical changes, and they are generally released by inflammation or damaged tissues. So when there is pathogen damage or a cut in the tissue or damage to the tissue, then we increase blood flow to those areas to deliver the immune cells or the white blood cells. We want to do that to get blood flow to those areas so that the white blood cells can start to clean up and flag the immune system as to what actually caused that damage. So histamine is the major change there. We also have a stretch response in the arterial walls. This is an autoregulation, and this makes sure that the arterioles don't get overly stretched. So when blood pressure changes are higher than normal, this makes sure that it will resist too high of pressure. So despite the increased pressure, it will normalize the flow to the tissues. This is most pronounced in the brain and the kidneys, where the damage of high pressure would be most severe. So this is called autoregulation, and it's a muscle response where when the vessel gets stretched too much because of high pressure, it will resist that stretch by contracting the muscle. This makes sure that the blood flow is kept constant, particularly in the brain and the kidneys. Another one is shear stress. This is internal stress on the endothelial layer, the inner layer of the blood vessels. And if we have extremely high flow in a particular vessel, then they will release nitric oxide. That causes vasodilation, which will then decrease the resistance and any potential damage that could be caused by this shear stress. Temperature. Temperature is another thing that changes with high metabolic activity. So when you have high metabolic activity, you have high heat. So warm tissues tend to vasodilate. So heat causes vasodilation, promoting increased blood flow to the tissues. What happens when we get cold, right? We sort of get pale and sometimes even sort of like turn blue, right? So cold actually causes vasoconstriction. That can help to counteract inflammation and histamine release. Those were all local changes. So now let's talk about changes to large groups of arterioles or extrinsic control. So this will change all of the arterioles in the whole system, not just a single group of arterioles at a particular tissue. So the sympathetic nervous system can supply the arterial smooth muscle, and as it releases norepinephrine, it will cause constriction or contraction of the smooth muscle and decrease that radius. Because this is extrinsic, it's going to happen everywhere except in the brain. When you do that, when you decrease blood flow everywhere by increasing resistance, then you're going to basically cut off the blood flow to the tissues that don't need it and maintain blood flow to the brain. One piece that you guys are going to struggle to understand is that when this happens, when the sympathetic nervous system causes vasoconstriction and cuts off blood flow to the tissues, it's going to increase blood pressure overall in the whole system. So what you're doing then is you're maintaining blood pressure, maintaining blood flow to the brain, and increasing the total systemic blood pressure. So how does that happen? I'm going to show you an equation, but you also just may want to memorize this. Local vasoconstriction cuts off blood flow, decreases pressure locally. Systemic vasoconstriction throughout the whole system diverts blood flow to the brain and increases blood pressure in the whole system. This is due to changes in the radius and resistance of the arteriole, so we can go back to our flow equation. Flow is the change in pressure over the resistance, and we can now rearrange that. If we want to look at the pressure, or the change in pressure of the whole system, we can rearrange this by multiplying resistance over to the left side of the equation. And what we get when we rearrange it is that the change in pressure is equal to the flow times the resistance. So as the systemic whole body resistance increases, then the systemic whole body pressure will increase. In other words, if all of the arterioles in the whole system are constricted by this extrinsic change through the sympathetic nervous system, the pressure will then overall increase in the whole system. We can then rearrange this equation and rename it. Awesome, right? Okay, so take this equation. Change in pressure is equal to the flow rate times the resistance. And now we have some new names that apply to systemic pressure. And we're going to call the systemic blood pressure now, instead of the change in pressure, we're going to call that the mean arterial pressure. We're going to call the flow now the flow through the whole system, which is also cardiac output. So the total flow through the whole body is cardiac output. And then the resistance, instead of being in an individual vessel, is now going to be the total body arterial resistance, or the total peripheral resistance. So the systemic flow then will be the total flow, the total cardiac output, the total pressure will be the mean arterial pressure. So now the equation to memorize is mean arterial pressure, or blood pressure in the body, is equal to cardiac output times resistance, or total peripheral resistance. We can also have extrinsic control through hormones. Mainly, this hormone is epinephrine and norepinephrine, the same molecule that was released from the sympathetic nervous system, but now it's going through the whole bloodstream through the adrenal medulla. So same, right? Sympathetic nervous system releases norepinephrine, adrenal medulla releases epinephrine, norepinephrine. Either way, the blood vessel walls have receptors for epinephrine, norepinephrine, and this will cause vasoconstriction. So whether it comes from the neurons, from the sympathetic neurons, or whether it comes from the adrenal medulla, epinephrine, norepinephrine is same, and you're going to get vasoconstriction. Because this is in the whole blood, you'll get vasoconstriction in the whole body, and that's why we call it extrinsic control. We have other hormones that can do this as well. ADH can cause vasoconstriction. Angiotensin II can cause vasoconstriction. And we'll talk about these more when we get to the kidneys. Okay, so let's put this together and draw this out a little bit so that we just have an idea of what's going on. So first, vasoconstriction is going to be increased contraction of the smooth muscle in the arterioles. When we do this, that will increase the resistance and decrease the flow through that vessel. So, vasoconstriction is going to be small. It's going to be a smaller radius. Smaller radius is going to increase resistance. If we increase the resistance, we're going to decrease local flow. But if we have vasoconstriction through the entire system, then that is going to increase blood pressure. Okay. So locally, we will have smaller radius, more resistance, and less flow in less active tissues. That is totally different than the entire system during stress and exercise, where the whole system vasoconstricts to increase blood pressure in the whole system. And that's because you divert blood to the muscles and the brain. Okay, so we're saving the blood flow to where it really needs it, and that overall increases the blood pressure in the whole system. Vasodilation then is going to be caused is going to cause large blood vessel radius. So these are large radius. This is going to be decreased resistance, and that is going to increase local flow. That is going to happen in active tissues, where you will see everything listed here. Low oxygen, high carbon dioxide, high nitric oxide, histamine release during inflammation, and then less sympathetic stimulation. So basically, metabolic activity is going to vasodilate locally. Okay. That's very different than during low stress or sort of, the rest state of the body, where if the vasodilation happens in the entire system, then that decreases blood pressure. And that is going to then allow increased flow to whole tissues. For example, the digestive system, where we can do the rest and digest activities. So the more active tissues will have local flow due to the increase in local metabolites. The less active tissues will have vasoconstriction due to the decrease metabolites. But that's totally different than the vasoconstriction that happens during stress, which overall increases blood pressure in the entire system. There's also vasodilation that happens during a lack of stress or during the rest and digest, which decreases blood pressure to the whole system. I know that's confusing. I promise it's going to continue to be confusing. So you need to memorize local versus systemic effects and take a moment to organize this for yourself. So here's a summary of control of the arterial radius. Local controls can be metabolic, histamine, stretch, shear stress, temperature. These are going to change blood supply to a particular tissue or organ. Extrinsic controls are going to be changes to the whole body or the entire system of blood vessels due to epinephrine, norepinephrine, either from the sympathetic nervous system or from the sympathetic nervous system stimulated hormone release. There's a summary diagram in your textbook as well, which talks about the changes in radius and their effects on total peripheral resistance. Okay, this is a good place to take a break, review so far what you have learned about the arterial regulation of vasoconstriction and vasodilation, the effects on blood flow and the effects on blood pressure. And now we're going to move on to some changes in the capillaries and changes in the veins that can also affect blood flow and blood pressure. So first, for the capillaries. So we're going to have adequate delivery to the tissues because of the arterial regulation. Once the blood reaches the tissues, we need to have exchange at the capillaries. So this occurs through the movement of materials into the fluid surrounding the cells. The fluid surrounding the cells is the interstitial fluid. So what we want is to have movement of fluid from the capillaries into the cells so that the cells can get the oxygen that they need. We also need to get the fluid from the cells back into the capillaries to return to the heart. So we're going to think both about diffusion forces on water and bulk flow forces on water that are affected by blood pressure. So bulk flow is what is occurring in the capillaries. So bulk flow is influenced by pressure gradients and not concentration gradients. So there are two pressures that are going to be important here. blood pressures or hydrostatic pressures and osmotic pressures. And both the capillaries and the tissues are going to have these pressures. This is really complicated and it can get really confusing really fast, so we are going to draw this out. Okay, so what we have is pressures that are going to drive fluid out of the capillaries and into the tissues. The first out pressure is going to be the capillary hydrostatic pressure. In other words, this is blood pressure at the capillaries. So you're going to have fluid pushing out of the capillaries, from the capillaries into the tissues. So this is fluid movement that will push on the capillaries, and that push of the hydrostatic pressure is going to push fluid from the capillaries to the tissues. So that is out of the capillaries to the tissues. We have another out pressure that is the interstitial fluid if it has any osmotic pressure. So we have osmotic pressure of this interstitial fluid, or the ICF. So this is solute pressure. So what you want to ask for this one is, do we have a high percentage of solutes in the interstitial fluid that can pull fluid out of the capillaries into the tissues? So in this case, again, it's movement from the capillaries to the tissues because of solutes that are in the interstitial fluid. This will only happen with pathology. So it is not normal to have high protein in the interstitial fluid. Why? Because the lymphatic system makes sure that the interstitial fluid is clean. So this out pressure is only if proteins are high in the interstitial fluid. And this is rare. So these are our out pressures. At the arterial end, so in the arteries versus the veins, we're going to have different blood pressure in the capillaries at the arterial end and at the venous end of the capillary. So at the arterial end of the capillary, we're going to have a blood pressure that is approximately 37 millimeters of mercury. If you go back to the very beginning of the lecture where we looked at the blood pressure in each of the vessels, that graph that traced everything, you'll see that it's about 30 to 40 or so by the time you get down to the arterioles and the capillaries. It's about 37 millimeters of mercury in the arteries and only about 15 millimeters of mercury in the veins. Oh, excuse me, 17 millimeters of mercury in the veins. So there's capillary blood pressure that is going to drive fluid from the capillaries to the tissues, and it is higher in the arteries than it is in the veins. We also want to know then, what are the pressures that would be driving fluid movement in to the capillaries? So that is going to be the interstitial fluid pressure, hydrostatic pressure. So this is basically fluid pressure that is present because of water in the interstitial fluid. Now, this isn't diffusion of water, this is hydrostatic pressure. So this is water that is pushing from the interstitial fluid to the capillary wall. And this will push from the tissues into the capillaries. The last inward pressure is going to be an osmotic pressure. And that is the osmotic pressure of the capillaries. So this is a solute pressure. Remember that water follows solutes, and this is going to drive fluid from the tissues to the capillaries. What this is the osmotic pressure of the proteins in the blood plasma. So remember that the proteins in the blood plasma are very high. So this is blood proteins that draw water in. So overall, we have fluid out and fluid in. And in order to get the net flow, we need to subtract the out minus the in. So let's look at the values then for the in. So we have 37 millimeters of mercury out at the arteries. And then for pushing back on the tissues, we're going to have a small amount of fluid pressure from the tissues to the capillaries. And this is because there is fluid in the capillary. So that's going to be about 1 millimeter of mercury. The osmotic pressure of the proteins in the blood pulling water from the capillaries in is much higher, and that's about 25 millimeters of mercury. So the dominant forces are the blood pressure the blood pressure out and the osmotic pressure in. If we then sum those up, and we're setting this osmotic pressure out to 0. So if we then add these, we're going to have 37 and 25 plus 1, and we're going to subtract those. So the pressure out minus the pressures in. So for the arteries, we're going to have out minus in is going to be 37 plus 0 minus 1 plus 25. So our net pressure at the arterial end is going to be 37 minus 26, 11 millimeters of mercury. So that's the difference between the out pressures and the in pressures at the arterial side. At the venous side, we have 17 millimeters of mercury. At the venous side, we still have very little solute pressure pushing out because there's very little proteins in the intracellular fluid. I should be saying extracellular fluid. Okay, you guys know what I mean, right? 17, 0. And then our pressures in, again, the fluid pressure from the tissues to the capillaries is not going to change much, just a little bit, so about 1 millimeter of mercury there. And then the solute pressure also not going to change much because the proteins stay the same. So that again is going to be 25 millimeters of mercury. So if we want to know what the driving force then is at the venous end, now we're going to take our out minus in, and that is going to be 17 plus 0 minus 1 plus 25, and our net pressure here is going to be negative 9. So what this says is that we had 11 millimeters of mercury moving out at the arteries, and 9 millimeters of mercury negative moving in to the veins. So in other words, we put this together, we have across the capillary to the tissues, capillary, we had net flow of 11 millimeters of mercury from the blood to the tissues at the arterial end. And we have a net flow from the tissues back into the capillaries of negative 9. That negative tells us it's moving in from the tissues into the venules. So this is doing exactly what we want in the body, which is to put the fluid into the tissues so that we can get the oxygen and the nutrients to the tissues, and then return the fluid back to the venules so that we can remove the waste and bring that deoxygenated blood back to the heart. So Overall, the four pressures we want to think about are the capillary blood hydrostatic pressure, that's the blood pressure in the capillaries, the plasma colloid osmotic pressure, that's the plasma proteins inside the capillaries, the interstitial fluid hydrostatic pressure, that's the fluid pushing against the capillary wall. And the interstitial fluid osmotic pressure, that's the low protein normally in the extracellular fluid. So this should normally not be the case, but there is some pathology if you have leaking proteins into the fluid that might cause problems there. So a net of 11 moving from the blood into the tissues, and a net of 9, negative 9, moving from the tissues back to the blood. This is how your book diagrams it, and it's exactly what we just said. We have our outward pressures minus our inward pressures. So out minus in at the arterial end gives us 11 at the arterial end. Out minus in at the venous end gives us 9 at the venous end. Is anybody bothered by the fact that the out pressure at the arterial end is 11 and the in pressure at the venous end is 9? Did anybody catch that? So if we're pushing 11 millimeters of mercury fluid into the tissues and we're only returning 9 millimeters of mercury to the veins, where does that other 2 millimeters of mercury fluid go? Well, that's the job of the lymphatics. So the lymphatic vessels are going to take that excess that does not get reabsorbed back into the veins and put it into the lymphatic system. So that 2 millimeters of mercury left over would pool into the tissues and cause swelling of the tissues if the lymphatics weren't there to pick it up. So the lymphatics are going to pick it up and make sure that it doesn't pool. Okay, before we get to lymphatics, I'm getting ahead of myself a little bit. So we have a change in the flow because of the change in the balance between the pressures. The pressure out at the arterial end is higher because of the basic change in the blood pressure at the arterial end to the blood pressure at the venous end. We can also control the flow of blood through the precapillary sphincters. You guys may have learned about these in anatomy class. This is smooth muscle that responds to local controls. So in addition to the bulk flow, we can control general flow to a region of tissue by opening these sphincters, that will get more flow, or closing those sphincters and then they will bypass that tissue. All right, back to lymphatic vessels. So we have this excess fluid. We had 11 millimeters of mercury moving out to the tissues and 9 millimeters of mercury moving back in from the tissues to the capillaries to be returned to the heart. The lymphatic vessels are going to take that excess fluid that was left over between the 11 and the 9 and return it back to the heart. Before they do that, they will also pick up any excess proteins, excess particles that are left in the fluid surrounding the tissues and make sure that it's clean and filtered before it gets back to the heart. So ultimately, we have blood flow to the tissues, perfusing the tissues and providing oxygen and nutrients to the tissue. Then the blood's going to return back to the veins to return back to the heart. Any excess fluid that is left at the tissues will be picked up by the lymphatics, cleaned, and also returned back to the heart through, do you guys remember where these enter in from anatomy? Subclavian veins. Yep, so they enter back into the subclavian veins, back to the vena cava to go to the right side of the heart. Okay, so we did arterioles and the major vasoconstriction, vasodilation effects on arterioles, both local and systemic. We did capillaries and the bulk flow that happens at the capillaries. The last bit before we finish up is to do venous return and then a little bit on control. So venous return is happening through the veins. Veins are low pressure, so blood will pool in the veins unless we help them out. So the blood pressure in the veins is only about 17 millimeters of mercury, and the blood pressure in the atria is 0. So this means that the lower the pressure in the veins get, the less blood will return to the heart. So if we need to help blood getting back to the heart to increase that delta P, increase that gradient, then we need to tighten up the veins. So there's several factors that affect venous return, and we're going to go through each one of these. First, we did this in the last lecture, but let's remind you. So when we increase venous return, or we increase the preload, or how much the heart fills, we get stretching of the atrial wall. So this is a cyclical change. When the atrial and the ventricular walls stretch, particularly the ventricular wall, we change the configuration of actin and myosin overlap. The better overlap we get, the better contraction we get. So when we stretch the ventricle wall, we get a bigger stroke volume and a bigger contraction. If we let more stroke volume pumped out of the heart, then ultimately the whole system increases in volume and we'll get more venous return. More venous return, bigger stroke volume. So it is cyclical. Okay? So the muscle wall will stretch. That stretch is called preload. That stretch maximizes cross-bridge formation with the actin and myosin binding. And all of this is described by the Frank-Stalling law of the heart. Increased venous return causes increased stroke volume because of the effect on contractility. So this is all because the heart acts at suboptimal cross-bridge length. So as the heart gets more and more full, as the heart stretches more and more, in other words, increasing the end diastolic volume or the filling, we'll get more and more output or stroke volume. More and more output will then increase down the line venous return. There are other effects that can increase venous return in addition to the effects on contractility and stroke volume. Sympathetic nervous system can increase venous return because we actually do have a little bit of innervation to the veins. So the veins have smooth muscle, and you can tighten up or constrict the veins, and that will actually help to increase venous return. That's because normally the veins are kind of pooling, they're kind of slack. And if you tighten them up, then it will increase the tone. This is very different than the vasoconstriction we just talked about. Venous vasoconstriction, in total contrast to what happens in the arterioles, actually increases flow because you went from this very slack, large vessel to something that has more tone. It doesn't affect the resistance like it does in the arterioles because veins are so much bigger. So, increased sympathetic nervous system increases vasomotor tone and gets more venous return back to the heart. We also have a similar idea, skeletal muscle pump. So if we squeeze on the veins a little bit more by squeezing the muscles surrounding the veins, then that will help to pump blood back towards the heart. Then we also have a respiratory pump. So this is because as the chest expands and decreases in pressure, it will increase the pressure gradient from the veins to the chest cavity. We'll learn about this a little bit more when we talk about ventilation, but suffice to say that the changes during breathing cause a pressure gradient that pumps blood and returns blood to the heart. So more breathing, more expansion and retraction of the chest causes increased venous return. So all of this is aided by the fact that veins have valves. So as we squeeze on the veins with sympathetic nervous system input, as we squeeze on them with skeletal muscle contraction, we're going to push blood. And we don't just push blood around in the veins, we push it towards the heart because the valves make sure that blood doesn't go backwards. So we're going to push on the veins and the valves make sure that it pushes towards the heart. There's also an effect called cardiac suction, and this is during ventricular contraction, as the AV valves are closed, they're actually drawn downward. That opens up the size of the atria, which creates a pressure gradient. So when you increase the volume of the atria, that's going to suction blood towards the atria. Last is you can affect venous return by affecting blood volume. We'll talk more about this when we get to the hormones of the urinary system that are affected by the kidneys, but the bottom line here is that if you increase salt and you increase water in the body, or both at the same time, then you can increase blood volume. More volume in the blood overall means more volume gets returned to the heart as the veins reach their maximum storage capacity. So increasing blood volume increases venous return, and this is a major way that the body increases venous return back to the heart. So overall, many ways to change venous return. The skeletal muscle pump, the respiratory pump, increasing the cardiac suction effect, the valves help to prevent the flow, and then also sympathetic nervous system input, which will increase the tone, increases in blood volume, which are going to happen through hormonal mechanisms. All of that will increase venous return. So let's pause for a second and think about all of the equations and factors that we have talked about today. And let's just back up and say, overall, we're thinking about the total blood pressure in the body because we want to know what is the effective blood getting to the tissues. And we're going to regulate these factors to make sure that enough blood gets out to the tissues. We can regulate mean arterial pressure, or the total blood pressure in the body system, by regulating cardiac output. We can affect cardiac output by affecting heart rate, which is sympathetic and parasympathetic input. We can affect cardiac output by affecting stroke volume. We can affect that by all of those venous return mechanisms we just talked about. We can also directly affect stroke volume by sympathetic input, which increases calcium and general contractility of the heart. And finally, we can increase the total blood pressure by increasing total peripheral resistance. That is done through the radius of the arterioles and vasoconstriction primarily. But you can also increase total peripheral resistance by making the blood thicker and by increasing the number of red blood cells. That affects viscosity. So go back to your resistance equation and you'll see that resistance is a function of radius, length, and viscosity. Okay, take a moment to try to digest everything we've talked about, or do this at the end. But this puts together everything we've just talked about as a summary diagram. So we'll put these equations together like this, and I want you guys to memorize these and be able to know how they fit together. So mean arterial pressure is the total blood pressure, which can be affected by cardiac output and total peripheral resistance. We can affect cardiac output by changing either stroke volume or heart rate, or both. We can affect the total peripheral resistance by changing viscosity or radius. Why don't we change the length of the blood vessels? Well, we have to actually grow new blood vessels to do that, and that's generally done during development, so that change is much more rare. So we can regulate blood pressure by changes in cardiac output and changes in resistance. Hopefully this is coming together for you guys. Just a couple more slides on how the brain drives these changes. So we actually have pressure receptors in the body. There are some located in the aortic arch, and there are some located in the carotid arteries. These pressure receptors are called baroreceptors. When blood pressure is not adequate, then the sympathetic nervous system will be triggered. It's triggered by the baroreceptors contacting the medulla. The medulla which is in the brainstem, has cardiovascular control centers. Those control centers will turn on and stimulate the sympathetic nervous system. If blood pressure is too low, the sympathetic nervous system will then increase heart rate and contractility. That will increase cardiac output. The sympathetic nervous system will also increase vasoconstriction, or total peripheral resistance, in the whole system. Remember, this is very different than a local change in vasoconstriction. Increasing resistance in the whole body increases systemic blood pressure, and this is all driven by baroreceptors to the medulla. If blood pressure is too high, the parasympathetic nervous system can decrease blood pressure, but it's much more limited. It can only affect blood pressure by decreasing heart rate. So this is one of the reasons that high blood pressure is so damaging to the body and dangerous, because we don't have a lot in our body that can counteract high blood pressure. So we can actually look at the sympathetic nervous system changes in arterial changes causing arterial pressure based on the input from the baroreceptors. So when blood pressure is normal, the baroreceptors are ticking away happily. If blood pressure is too high, lots of signals from the baroreceptors. If blood pressure is too low, then very few signals from the baroreceptors. And this overall is going to trigger the sympathetic nervous system effects. So if blood pressure is too low, then there are going to be action potentials in these baroreceptors. That's going to activate the cardiovascular center in the medulla. That will increase sympathetic, both effects on contractility and effects on heart rate and vasoconstriction. That will increase heart rate, increase stroke volume, and increase vasoconstriction. Overall, that will affect cardiac output, total peripheral resistance, and put blood pressure back to normal. All right, take a moment to now review the parasympathetic and sympathetic effects on blood pressure. Sympathetic can do much more than parasympathetic. Sympathetic can affect heart rate, it can affect total peripheral resistance, and it can affect stroke volume. Parasympathetic can only affect heart rate. Cardiovascular disease then accounts for over half of all the deaths in the United States. A myocardial infarction is the technical term for a heart attack, and this happens when the heart muscle does not receive enough blood. So if the coronary arteries around the heart are deprived of blood, they are blocked, then not enough blood gets to the heart tissue will die, and you will lose the connections which allow the heart to pump and fill for the cardiac cycle. Another important piece of cardiovascular disease is atherosclerosis. This is plaques of cholesterol and other substances that form inside the arteries and block the arteries. So overall, this blockage of the arteries is not only dangerous because it affects the arteries themselves, but also because these plaques can break off and form clots throughout the rest of the body. Okay, phew. I know that was a lot of equations for today. Hopefully you guys took some time to get them all down. Please let me know if you have any questions.